learners. I'm Dr. Daniel, HOD of Zoology, JKC College, Guntur, Andhra Pradesh. In my first video lesson, I explained about the introduction of cloning vectors, types of cloning vectors, and one of the types of cloning vectors, that is the plasmid vectors, especially PBR322 plasmid vector. In this lesson, this is my second video lesson, which I would like to introduce you or continue the cloning vectors. So in this lesson, I will explain about the PUC vectors, which are one of the plasmid vectors, and another type of cloning vectors, that is bacteriophage vectors. Okay, let us see. First, PUC plasmid vectors. Before I go into this lesson, let me know what is cloning vectors. What is cloning vector? Cloning vectors are the DNA molecules which carry the particular foreign DNA which we can insert or which have to be recombinated. So it is nothing but a DNA molecule. Say for example, PBR322 is a vector which carry the gene of interest which we want. So like that, all the cloning vectors, they can carry a foreign DNA molecule to the clone. Okay, this is the another plasmid vector which is called PUC. So you can see the contents of these plasmid vectors: nomenclature, characters, structure of PUC vectors, advantages of PUC vectors, and disadvantages of the PUC vectors. So let us see one by one quickly. Number one, nomenclature. So these plasmids are given the names of the places where they were discovered. Say for example, P, small p indicates the plasmid. UC stands for University of California, where it was first developed by a scientist called Messing et al. That means other scientists included. And there are different series of PUC vectors developed, and one of the best these PUC plasmid vectors are PUC8 vectors. So here I explain about this PUC8 vectors. So what are the main characters of PUC vector or PUC8 plasmid vectors? First, these are modified PBR322 vectors. And secondly, these are smaller than PBR322 vectors, having only 2.7 kilobytes. And third character is they have 2676 base pairs in size. And fourth character, comparatively, they have high copy number and produce 500 to 600 copies of plasmid per cell without amplification. So the producing of the particular number of plasmids is very high when compared to other plasmid vectors. Next, what is the structure of the PUC8 vectors? So it includes the following important parts. The first one is the ORI, origin of replication. It is derived from the origin of the PBR322 plasmid vector. The second one is the selectable marker. marker. So this marker is nothing but the ampicillin resistant site, but it doesn't have the tetracyclination gene or site which is present in the PBR322. So the transformed host cells can grow on the media having ampicillin, whereas non-transformed will die. That means in the ampicillin media, a transformed host cells, say for example E. coli, which contains the recombinant DNA, can grow only in ampicillin containing media. So that is the special feature of these selectable markers. And the third important feature or structure of PUC8 is the presence of LAC-Z gene. LAC-Z gene having MCS means multiple cloning sites, multiple cloning sites. So you can see the diagram or the diagrammatic representation. This is the PUC8 plasmid vector, which contains three important regions. First one is the AMPR, ampicillin resistance site, and this is the ORI, origin of replication, where the vector can replicate itself, and the LAC-Z gene, which contains so many restriction sites or the restriction enzymes to act, where we can insert the recombinant DNA or the gene of interest. So this is the diagrammatic representation of the PUC8 plasmid vectors. And here I would like to explain more about the MCS. What is MCS? Multiple cloning sites. Multiple cloning sites. So MCS is the part of the gene, that is the LAC-Z gene, which codes for a portion of an enzyme called beta-galactosidase. It codes for a portion of an enzyme called beta-galactosidase. What is the importance of this enzyme? Let me explain. When the plasmid is used to transfer the host cell, that is E. coli, the gene must be switched on by adding an inducer called IPTG, isopropyl BD1 thyrogalactopyrinoside. That is the abbreviation for IPTG. In the presence of this inducer or IPTG, the beta-galactosidase enzyme is produced. This functional enzyme is able to hydrolyze a colorless substance in which media the bacteria grows, called hexagon, into a blue insoluble material. That means if the beta-galactosidase enzyme is produced, automatically it can convert that media, hexagon media, into an insoluble blue colored substance. However, if the LAC-Z gene is disrupted by insertion of a foreign fragment of DNA, that means if you want to insert a particular gene of interest in the LAC-Z gene, the non-function of enzyme results. This is called insertional inactivation. It is called as insertional inactivation. When you insert a particular foreign DNA into that particular gene, or in the place of LAC-Z gene, automatically it will be inactivated. That means it won't produce that particular enzyme, which is called beta-galactosidase. So because of that, what will happen? The hexagon media, it remains colorless, or it remains white in color. This indicates that the recombinant DNA is already it has been produced in that particular media. Understand? So this is called blue-white screening of that particular vector, or to screen whether the DNA or recombinant DNA produced or not. If the recombinant DNA is produced in the media, means the color is white. If the recombinant DNA is not produced by the vector, that means the color of that media, it becomes blue in color. Okay. So what is that media? It is called X-gall media. X-gall media, it is nothing but 5-bromo, 4-chloro, 3-indole, BD, galactose. Galactose. So this is a very important phenomena which you have to understand when you study regarding the plasmid vectors. Okay. Now, what are the advantages? What are the advantages of these PUC8 plasmid vectors? The first one is easy screening due to blue-white screening selection. As I just explained about that, the blue-white screening, it is a very easy screen for selection. If you select these plasmid vectors, means definitely that experiment will complete earlier than other plasmid vectors, because the blue-white screen is the best method to calculate or to see whether the recombinant DNA is produced or not. And the second advantage or use is the property, they have the property of high copy number. That means one cell can produce up to 500 to 600 copies. And another advantage is due to the presence of this MCS, multiple cloning site, it increases the number of potential cloning strategies available. It increases the number of potential cloning strategies available. And these plasmid vectors also used as both cloning as well as expression vectors. Not only to produce a particular multiplication of gene of interest, but also they can produce the proteins also, or enzymes, the production of enzymes. Expression vectors means production of enzymes due to the expression of the genes. And these plasmid vectors are inexpensive. They are not as expensive or as other plasmid vectors. So there are so many advantages, which just I have given only a few over here. And what are the disadvantages? There are some disadvantages. For example, they show false positive and false negative results. Sometimes the results may be, if it is negative, then it shows positive, vice versa, like that. So that is one of the disadvantages. And another disadvantage is instability during copying. So the transcription, during the process of transcription, during formation of mRNA, it may be sometimes it is not in a stable condition during the copying. And it cannot accommodate a gene of interest larger than 2.7 K. Of course, it is advantageous than plasmid vector, but the accommodation, as far as the insertional accommodation is concerned, it is comparatively lesser. Okay, that is about the PUC8 plasmid vector. Now, come to another type of plasmid vectors. Sorry, another type of cloning vectors, that is bacteriophage vectors. This is the second type of cloning vectors which are used in the animal biotechnology. So first, see what is this? What is a bacteriophage? Bacteriophage means, is it a virus or bacteria? Bacteriophage is a virus. So generally, most of the students, of course, not most of the students, but some of the students, they think that bacteriophage means these are bacteria because this name includes bacteria, no? But these are not the bacteria, these are virus. So bacteriophage are the viruses that infect bacteria. First, it was first discovered by Frederick Twart and Herrely in France in 1970. So D. Herrely coined the term of bacteriophage, meaning bacterial eater, to describe the agent's bactericidal activity. That means this virus, it can eat the bacteria. It infects the bacteria means definitely it can take something from the bacteria. That's why it is a bacterial eater as far as Herrely is concerned. So these phages are very simple in structure. Mostly they consist of DNA, double deoxyribonucleic acid, and occasionally they contain the ribonucleic acid, that is RNA. Okay, it has or it undergoes two types of life cycle. So I will discuss later on. So there are two types of bacteriophage vectors. There are several bacteriophages, but the most commonly used are these are the two bacteriophage vectors. Number one, lambda phase vectors, and number two, M13 phase vectors. Let us see quickly one by one. So the lambda phase vectors. These were first discovered by Esther Lederberg. You see, this is nothing but virus, no? This is nothing but virus having one head, collar, and tail, and also some tail fibers to enter into the to bind to the bacterial cell. So, as I said, it is nothing but a bacterial virus. It infects generally the bacteria species, that is Escherichia coli, E. coli. We know very well about this Escherichia coli. So it has two types of life cycles. Number one, lytic cycle, and number two, lysogenic cycle. What is the difference? In the lytic cycle, the viral DNA exists as separate molecule within the bacterial cell, and it replicates separately from the post-bacterial DNA. That means it won't integrate with the nucleoid of bacteria or E. coli. It can separately replicate within the bacterial cell or within the cytoplasm of the bacteria. That is called lytic cycle. And the second type of lysogenic cycle is it completely integrates with the bacterial DNA. That means the viral DNA can integrate with the bacterial DNA and replicates along with that host DNA. So two types of life cycles. One is lytic cycle and another one is lysogenic cycle. Here you can see both lytic and lysogenic cycles. Here you can see this virus, it infects the bacteria and it enters into the bacteria where it reproduces, but it doesn't integrate with the main nucleoid DNA. It can separately reproduce in the cytoplasm by using the mechanism of bacterial cell, and after reproduction they will come outside. Whereas in lysogenic, this virus or bacteriophage, it integrates with the bacterial DNA, then it combines with the DNA of the bacteria. From there they replicate and they will come outside. So out of these two, the lytic cycle is a very important cycle to use in the cloning purposes. Okay, lysogenic cycle is not necessary for us, but whereas the lytic cycle is essential for the replication of these bacteriophage vectors. Okay, how it penetrates? It is general description. This binds to the target E. coli cell by the tail fibers, and then it enters into the bacterial cell. And what are the main features? Lambda phase vectors or lambda phase bacteriophage vector contains the following features. Number one, they have approximately 49 kb, 49 kb, more than 48,000 bps base pairs. And the second point is these are linear with cos sites in the head and become circular after it enters into host along with recombinant DNA. First, it may be linear or not circular, straight. So soon after entering into the host cell, it becomes a double-stranded DNA, which consists of the recombinant DNA, which we are going to introduce. And the third feature is this vector consists of three important parts. Number one, ORI, origin of replication as usual, as you have seen in the plasmid vectors. Two selectable markers. One is ampicillin and another one tetracycline. Okay, and the third one is cos site. Cos sites are very important sites for the ligation. Okay, and another feature is the lambda phase replicates in two types of life cycle, that is lytic and lysogenic, as already I explained that. So where we want only the lytic cycle. These are the important features of lambda phase vectors. So these are the examples, two examples for, of course there are two types of vectors. Number one, insertional vectors. Two types of vectors are there. Number one, insertional vector, and another one is the replacement vectors. In the insertional vectors of lambda phase vectors, a part of DNA which is non-essential, is present. So in that place we can include the gene of interest. Say for example, you can see here, this is the non-essential region of insertional vectors. And example for insertional vector, lambda GT10 and lambda GAP11. And of course, these are the descriptions. And the second type of vectors is the replacement vectors. So these are nothing but lambda phase vectors in which a part of DNA, a part of segment of DNA can be deleted. In that place, we can include the part of gene of interest. Here you can see the LAGZ gene. Here you can delete this particular part, and there we can introduce the gene of interest. It is called stopper gene. The deleted gene is called stopper gene. So these two types of vectors are there. Number one, insertional vectors, and number two, replacement vectors. Okay. And the example for lambda EMBL4 is, it is nothing but the example for replacement vectors. Okay, learners, I will conclude over here. So you can do, or you can watch my YouTube channel, Dr. DK Zoology YouTube channel. Okay. Thank you very much. Thank you, one and all.